Λοιπόν, καλημέρα σε όλους από τη Βοστόνη. Είναι περίπου μισάνυχτα της Παρασκευής και ζητώ συγγνώμη που δεν μπορώ να είμαι εκεί απευθείας. Αλλά είμαι πολύ χαρούμενος που έχω την ευκαιρία να σας μιλήσω και ευχαριστώ πραγματικά τους οργανωτές που έχουν οργανώσει μια τόσο όμορφη συνάντηση και που με έχουν καλέσει και εμένα να μοιραστώ μερικές από τις δικές μου δουλειές. Θα δώσω την υπόλοιπη μου ομιλία στα αγγλικά, ελπίζω να είναι εντάξει. So what I'd like to tell you about today is our work on trying to decipher and manipulate human disease circuitry at single-cell resolution, and specifically how artificial intelligence can really make a big impact in understanding human health and also enabling the future of medicine. So my name is Manolis Kéllis. I'm a professor in the Massachusetts Institute of Technology at MIT, and also a member of the Broad Ινστιτούτου and of the Computer Science for Artificial Intelligence Laboratory at MIT. So I'm going to start with the story of human genetics. Human genetics goes back thousands of years with the selective breeding of animals and plants, and the realization that eye and hair color inheritance are passed on from parents to offspring throughout human generations. But it's not until 1860s that Gregor Mendel figured out the rules of inheritance, and specifically the discrete nature of inheritance, that there is no blending, that there are dominant and recessive alleles that are independently assorted from generation to generation. This information was used to start mapping the position of these strong effect Mendelian traits along the lines of the chromosome based on their co-inheritance patterns. And in the 1980s, this led to extreme success in being able to map dozens of Mendelian traits with strong effect alleles. But the continuous nature of human variation was not reconciled with this view until the beginning of the 19th century, where Ronald Fisher and other statisticians finally were able to show that even a small number of Mendelian traits could actually explain a seemingly continuous variation, such as for human height. And here we enter the early 2000s, where the human genome is first sequenced. We can now finally start building variation maps that systematically catalog human variation and then build haplotypes, namely blocks of co-inheritance in the human genome, and then use those to enable genome-wide association studies to look at how genetic variation correlates with phenotypic variation. You can think of Mendelian genetics as hammers hitting individual genes in the genome and knocking them out. By contrast, complex genetics is about thousands of needles that are perturbing the system ever so slightly. So these are common variants that have very weak effects, and understanding their function has been a huge challenge. So the work of my group seeks to start with human disease genetics of common variants and rare variants and understand their molecular mechanism and their role in disease. Here's an example of body mass index. This is a genome-wide association study. What we call this is a Manhattan plot based on the tall skyscrapers that are hanging up above all the other genome-wide significant loci. And you can see here the FTO locus, which was promptly renamed Fat and Obesity Associated, reigns supreme among all others. So the question is, how can we understand the mechanism through which this locus works? How can we systematically translate these incredibly powerful findings of genetics into mechanistic insights? And the way that we do that is that we start with genetics and then we profile across thousands of healthy and disease samples from many individuals the RNA, namely the expression of the genes, and the epigenome, namely the accessibility, the DNA histone modifications, or the DNA methylation of the genome. So the RNA tells you about the quote-unquote output of how a gene is expressed and how it functions. And the epigenome tells you about the upstream regulation, the circuitry, if you wish, for how that gene is wired. And this wiring, this circuitry, it turns out underlies 93% of the genetic variants. For 93% of cases, genetic variants that are associated with disease do not perturb the proteins directly. Instead, they act to modify gene regulation, to alter either the accessibility of the histone modifications or the DNA methylation or some function of the epigenome and ultimately affect RNA expression. So the question is, how do we now use, instead of these large hammers, how do we understand these thousands of needles? So we now first understand their molecular effects, and then we need to integrate the data computationally to understand how to predict what are the drivers. How do we understand the language of the DNA so that we can interpret individual mutations based on the impact that they have on those driver regulatory motifs? So this is all about understanding the language of DNA. It's all about programming. It's all about computer science. And that's why I hope a lot of you are feeling very comfortable with this concept that if we find differences in this code and we can see how these differences are changing the intermediate program, then we can kind of understand this language, understand how to predict driver genes, genetic regions of control, the circuits, and the cell types where they act. So that's what we do systematically. We basically develop a lot of Bayesian methods, deep learning methodologies, graphical neural networks, and convolution neural networks, and models of causality of Mendelian randomization to try to understand the mechanism through which these genetic variants are acting. And once we have these predictions, we then seek to validate them systematically. To understand the causality, we start with genetics, because genetics has a causal component, but to show that whatever we infer is indeed causal, as downstream consequences, we then go and directly manipulate the corresponding genes, regions, and cell types to validate that indeed our predictions of causality are real, and that we can indeed manipulate these circuits in human cells and in animal models systematically. We then disseminate the results and start the cycle all over again. So the first step is this large-scale data generation. So we have not only a computational component in my laboratory, we also have an experimental component. And we have profiled thousands of human samples at single-cell resolution, understanding Alzheimer's, frontotemporal dementia, Lewy body dementia, ALS, Huntington's disease, psychosis, schizophrenia, bipolar disorder, Down syndrome, autism, post-traumatic stress disorder, addiction, aging, cardiac disease, obesity, and exercise, and how all of those affect the phenotype, the molecular phenotype, these intermediate molecular phenotypes of disease in concordance with both genetics and environmental effects, to then ultimately lead to the observed functional consequences on the physiology and the biology and the cognition of human individuals. So again, we're looking at both DNA accessibility through single-cell assay for transposes accessible chromatin or single-cell attack, and through single-cell RNA profiling, very often in multiple regions of the brain and in multiple tissues in the body. The second step is to integrate all these data sets. For example, we can use these genome-wide statistics of the enrichment of different epigenomic annotations across different cell types and how they enrich in the genetic variants that are associated with different traits through the linkage disequilibrium patterns of co-inheritance of different genetic variants, and how this genome-wide empirical prior through these enrichments can basically be used to reweigh the evidence that we have from individual epigenomic annotations into a posterior probability of a causal indicator variable that basically tells us how likely is it that this particular genetic variant is indeed the causal one on the way to disease. We also carry out a lot of mediation analysis and causality inference, basically understand whether the genetic effects on the phenotype are in fact mediated through intermediate molecular phenotypes that allows us to now paint a path of causality. One approach for doing that is, instead of just looking at their correlation between genome-wide methylation and genome-wide and the disease output, similar to genome-wide association studies for GWAS, we can basically look at methylome-wide association studies, but that is a bidirectional arrow because the disease could be influencing the methylation rather than the methylation influencing the disease. So instead, what we're looking at is predicting genetically the genetic component of methylation rather than all of methylation. So this imputed methylation, when correlated with disease, now has a unidirectional arrow, because we have causality back, because this is only the genetic component of methylation. which is unlikely to be driven from the disease itself. The third type of approaches that we're using is convolutional neural networks, where we can predict from DNA sequence an alphabet where the filters, these building blocks, these primitives of the lower layer of convolutional layers, in fact, match known regulatory motifs that we can then learn de novo. And by combining multiple of these motifs, we can start understanding through multitask prediction the accessibility patterns of different cell types, which allows us to now start asking what will be the impact of perturbing this T or this C in a new cancer sample that has never been observed before, or in an individual that carries a rare variant. So by being able to start predicting function directly from sequence, we can now start predicting the impact of these mutations. And the last approach that we're using is geometric deep learning, being able to not only understand these convolutional layers, but being able to sort of pay attention to specific parts of the chemical space or the protein space or the structure of single cell expression data to try to understand the relationships between different types of elements in a much more structured way. So we're applying this geometric deep learning to both single cell RNA, but also to chemical biology and protein structure. And I'm going to talk a little bit more about that at the end. So what do these steps give us? These two steps are basically giving us now the ability to understand the circuitry. So in the case of FTO, the strongest genetic association with obesity that I showed you earlier, what we were able to show is that the FTO gene itself has nothing to do with obesity. Instead, there's a regulatory motif that sits within the first 10,000 letters of the intron of FTO, where the association lies with obesity. And within these 10,000 nucleotides, there's a single letter change that changes an AT-rich motif that is normally bound by AT-rich interacting domain factor ARID5B. This motif is normally bound by ARID5B and represses this 12,000 nucleotide promoter that basically allows, or super enhancer, I should say, that allows you to now control the activity of these two genes downstream, IRX3 and IRX5. So these genes were previously not known or not implicated with obesity, but what we showed is that they are the true targets of this individual letter change and this upstream regulator through this super enhancer, and that they act specifically within adipose tissue. So IRX3 and IRX5 are not adjacent. IRX5 sits 1.2 million letters away, and IRX3 sits 600,000 letters away, several genes over from this genetic association. That basically means that we're really tracing the circuits rather than just looking under the lamppost. So these genes we showed are in fact master regulators of thermogenesis or heat generation that allows you to burn excess calories in your diet instead of choosing to store them as white adipocytes in the form of lipids. So lean phenotypes occur when you're able to burn these excess calories, and obese phenotypes are able to occur when you're not able to burn them, when you store every calorie that you consume. So why are we excited about these circuits? Not because we're academics and we want to sort of figure out how they work. I mean, of course, we care about that. But much more importantly, because by having the circuits, we now know how to manipulate these circuits. So we now have knobs that we can intervene on. We can increase or decrease the expression of the upstream regulator, increase or decrease the expression of the downstream target genes. And we can even go in these individual letter that we predict to be driving this association, and then change it back and forth from C to T to restore lean phenotypes by restoring the AT-rich motif and then restoring the repression of IRX3 and IRX5 by the upstream regulator. Or conversely, by shifting from lean to obese phenotypes. So just to give you a few of these examples, we basically have the ability to restore thermogenesis measured as oxygen consumption rate, both in basal and in isoproterenol stimulated conditions. So what we show is that individuals who are homozygous for the obesity risk allele are now suddenly able to burn these calories in their white adipocytes. So taking primary white adipocytes from risk individuals, we now are able to restore the normal function of these cells and be able to again burn calories through thermogenesis, through this depolarization of the mitochondrial membrane. And this is now at the level of a single letter. What about at the level of the downstream target genes? Because evolution will only tolerate small effect changes of these letters. But the downstream target genes, we can intervene much more severely than what the single letter alterations would allow us to do. So what we can do is actually go into mice and repress these genes that get de-repressed in thermogenesis, in obesity. So by repressing these genes, we hypothesize that we'll be able to sort of repress this pathway of white adipocytes and in fact force the cells to constantly burn all of their white fat. And this is exactly what we see when we put mice under this dominant negative construct for IRX3, under the control of an adipose promoter. What we basically see that the mice are leaner to start with, but very importantly, when we introduce a high-fat diet, these mice are normally gaining weight, but in this construct, they're unable to gain weight. And if you open up these mice, you basically see that all of their fat stores are completely red. There's no white fat. They've simply burned off all of these calories, and there's no white fat storage. It's all brown, red, healthy tissue. So this is what we want to do systematically across the human genome. So this is the strongest genetic association with obesity. We also looked at the strongest genetic association with Alzheimer's disease. We took male and female individuals that were carriers of ApoE3, ApoE4, and heterozygous ApoE3/4 carriers. So this is apolipoprotein E, which is the strongest association with Alzheimer's. If you carry 2 copies of ApoE4, your risk for Alzheimer's is between 8-fold and 12-fold higher than the rest of the population. So you really don't want to be carrying that. So you want to know, if I am a carrier, what can I do to change my phenotype? So we basically asked, what is the difference between carriers and non-carriers, whether or not they have Alzheimer's disease and whether they're male or female? And what we found in all cases, even before Alzheimer's and of course after Alzheimer's, is that ApoE4, this genetic variant, is in fact predicted through our single cell data and through this computational analysis to in fact impact cholesterol transport. What does cholesterol do? Cholesterol is used by the myelin of the oligodendrocytes, these myelin-creating cells. Cholesterol is used to form the myelin and to protect the neurons. So what is the myelin? It is basically this lipid sheath, this cholesterol-based sheath that is allowing the neurons to be protected from the environment and to fire much more accurately. And without ApoE3, with ApoE4 individuals, we see that many of these cells are lacking these very nice, thick, protective barriers. And it turns out that if we restore cholesterol transport, then the ApoE4 individuals are able to restore their myelination. When we look at ApoE4 cells in a dish, we see that they restore myelination. And if you look at mice, we see that the mice restore myelination, and also restoring the cholesterol transport actually restores cognition itself in the mice. So again, this is what we want to do systematically. We want to systematically manipulate human disease circuitry in order to be able to reverse these phenotypes. And we want to do this across thousands of loci in the genome. So we basically scaled up from 127 epigenomes that we had profiled in Nature in 2015 to 834 epigenomes in a paper that was published in Nature in 2021. And what we basically showed is that we can now identify systematically the genetic traits that are enriching in specific tissues. So we can now start building a network that basically tells us which genetic traits are enriched in what tissues. And what we're finding is that if you look at cholesterol, it enriches very specifically in the liver. If you look at educational attainment, intelligence, cognitive performance, but also smoking, depression, worry, neuroticism, these genetic traits cluster very strongly in enhancer regions, in gene control regions that are active in the brain. If you look at kidney, you basically see all these filtering functions. And if you look at Alzheimer's disease, what's really surprising is that Alzheimer's is actually clustering not with the brain, but instead with these immune cells. And we published a paper in 2015, also in Nature, that basically argued that, in fact, there's probably the primary driver of Alzheimer's disease is probably having to do with myelination and with these immune cell clearances that are involved in all of these immune functions of the body. So we have since went from these tissue level understanding to a single cell level understanding. So in a paper also in Nature in 2019, we basically showed that if you now take 48 individuals with that are normal phenotypically, without any pathology associated with Alzheimer's disease, no neurofibrillary tangles, no amyloid. If you just look at cognition, they're very highly cognitively capable. And then you can take 24 non-AD individuals and 24 AD individuals, 24 Alzheimer's individuals. And what we're seeing is that these Alzheimer's individuals are showing dramatic changes in their single cell profiles. Here I'm showing you 84,000 cells from these 48 individuals. Every one of these dots is a 20,000 dimensional vector. And we're using T-statistics stochastic neighbor embeddings. So these two-dimensional, highly nonlinear embeddings that allow you to take these 20,000 dimensional vectors and project them down into only 2 dimensions, which I'm visualizing here. And you can see that in this projection, the major cell types are in fact highly distinct from each other. Moreover, if you look at the presence of amyloid that is low or high, or different stages of pathology or cognition or amyloid variable, then you basically see that major clusters in these cells are in fact identifying differences phenotypically between different individuals. And we can also look at somatic mutations. We can also look at the accumulation of individual nucleotide changes in the transcript that are not there in the germline. And what we see is that these accumulations are in fact clustering in very specific biological processes, indicating that somatic burden might in fact be contributing to disease as well. We have since scaled from 84,000 cells in this picture to 1.6 million cells. We looked at 7 different brain regions across 48 individuals, looking at the prefrontal cortex, midtemporal cortex, angular gyrus, entorhinal cortex, thalamus, hippocampus, and mammillary body. And for each of those brain regions, profiled 48 individuals, 24 cases, 24 controls for Alzheimer's. What do we see? We basically see that we can discover several dozen different neuronal subtypes across these different regions that we can then start classifying based on their marker genes. Here, I'm showing you 30 different subtypes of excitatory neurons, and we have another 23 subtypes of inhibitory neurons. This allows us to actually start seeing how these change across different cell types. And very importantly, having 1.6 million cells computationally allows us to now start predicting where in the time course of Alzheimer's progression every cell lies. So we can take transcriptional signatures, gene expression signatures that are indicative of Alzheimer's or of control cells, and then start predicting for every one of those cells how Alzheimer's-like or how control-like that cell is. And we're also finding many intermediate cells. And what we can do is actually start tracing the path of Alzheimer's and start asking for every gene associated with Alzheimer's, where does it sit on this path? How can we predict the early acting, middle acting, and late acting genes underlying Alzheimer's? We have also gone a step further and looked at the anatomical organization of the brain. So if you look at the hippocampal formation, you can see here the dentate gyrus, CA1, 2, 3, subiculum, and then the different layers of the entorhinal cortex. What we can see is that these cells have very different morphologies, and those morphologies are associated with distinct transcriptional profiles. So the gene expression profiles, again, when projected from this 20,000 dimensional vector into this two-dimensional vector, are in fact showing distinct clusters that are separating cells with different properties from each other. And they're in fact recapitulating the spatial organization of these cells. And we can use that to start distinguishing the association with Alzheimer's of dentate gyrus or CA1 and show just how dramatically different they are in their predictive power. We have also used the technique known as spatial transcriptomics. That's basically looking at these pucks with probes that allow you to now mark all of the cells that are under your puck with the same barcode. And then when you sequence these cells, you can kind of place them back into the two-dimensional organization of these tissues. But this gives you pseudo bulk. This basically gives you mini bulk experiments, which are containing multiple cells at a time. And we've developed algorithms to be able to deconvolve these multiple cells into individual cell types that are much more distinct phenotypically. And that allows us to now start recognizing the differences in the proportion of Purkinje cells, for example, which is a rare cell type in the cerebellum, which is affected during this ataxia, this CA1 disease. We've also developed algorithms for bridging the translation table between the spatial organization of cells and their single cell profiles, by basically training these deep learning models to predict the spatial position of individual cells from their expression patterns alone. And we've also looked at subcellular resolution, where we can now look at thousands of marker genes and then ask for every one of those marker genes, where is that gene expressed in the neighborhood of each individual cell? And where is it expressed relative to, for example, here, the vessels, the blood vessels that are basically irrigating the heart or the brain. So there's a huge room for developing new computational methodologies in this space. And again, if you're interested in both spatial organization and single cell data sets, then this is a very, very important field. We've also developed methods for integrating genetic findings with electronic health records. We've looked at hierarchical models that allow us to capture the phenotype frequency, the lab measured frequency, and the binary clinical features, and now decide if there's a missing lab test because the doctor didn't prescribe it. What does that tell us about the underlying distribution of the values for those missing data? And we basically developed this non-missing at random model called Mixer for Mix EHR for electronic health records that allows us to now learn from these clinical records in a multimodal way between lab tests, ICD-9 billing codes, prescriptions, DRG codes, and doctor notes that allows us to now extract information jointly and have these features help reinforce each other. And we can go from a very sparse clinical record to a very highly populated clinical record. And we can start subdividing individuals into subtypes based on the sets of variables that they have in their clinical record. And we can now start correlating these groups of individuals with genetic variables that these individuals carry, and then predicting from expression quantitative trait loci or EQTL analysis, the cell types where these genetic variants are predicted to have the strongest effect on the expression of neighboring genes, to start predicting what are candidate causal driver cell types underlying these subgroups of individuals. Beyond these electronic health record phenotypes, we've also looked at transcriptional phenotypes. So we call these transcriptional hallmarks, and this is inspired by the hallmarks of cancer that Bob Weinberg, for example, has laid out. It basically says that there's a certain set of pathways that underlie cancer. And in the same way, what we're trying to do is discover de novo the individual pathways that underlie Alzheimer's disease. How? By looking at 430 individuals here along the columns, and then for every one of those individuals, ask what are the transcriptional dysregulations that are happening for those individuals. And if we see that there are, in fact, groups of dysregulations that are happening recurrently across multiple individuals for the same cell types, then that allows us to now start predicting what are these transcriptional hallmarks. And to annotate these hallmarks, we basically ask what are the sets of genes that belong in these hallmarks, giving us distinct pathways that are associated with each of those. So we can find, translation, oxidative phosphorylation, synaptic biology, zinc and metal ion, GTPase regulators, and so on and so forth. But from those distinct pathways, we can now start painting every individual in a distinct way. So we can use these transcriptional hallmarks to infer distinct patient subgroups that allows us to now start reasoning about which patients contain what transcriptional regulations and start associating those with phenotypic differences between these patients. And we see that, for example, education, cognition, post-motoral interval, pathology, Bragg stage, etc., are in fact differentially associated with these transcriptional hallmarks. And that allows us to now recognize subsets of individuals that have distinct phenotypic signatures. What you can see here is that hallmarks 8, 10, and 11 are rarely associated with late-stage AD individuals, but other hallmarks are very much associated. So that basically tells us that perhaps these hallmarks are indicative of patients who are protective against Alzheimer's disease. And we can now start using the genetics of these individuals to start predicting the transcriptional hallmarks of every person, even though we do not have access to their brain. So we just need a blood sample. And from that blood sample, we can infer the genome-wide genetic differences of those individuals, and then use those genetic differences to then go in and predict the transcriptional dysregulation of every person and how that leads to a grouping within different treatment groups. We've also developed these Bayesian models for understanding the relationship between biomarkers and mortality or other outcomes in clinical records based on the longitudinal nature of these records and how individual patients vary across different biomarkers. And what we observe is that there's both a positive and a negative correlation between these risk biomarkers and the patient outcome. The positive comes because the observed biomarkers are informative of the underlying risk for the disease. But the negative correlation comes from the fact that when the doctors observe these biomarkers, they're much more likely to provide a stronger treatment that will then give better outcomes to the patient. And this is one of such examples for creatinine. If you look at creatinine levels in the blood, basically see that at exactly the value of 3, the risk suddenly decreases. And at exactly the value of 5, the risk again decreases. What's going on here? What's going on is that there's a monotonic relationship between creatinine level and risk, true risk. But in fact, when the doctors see that the creatinine levels are rising, they administer one change at a given threshold and a different change at another threshold. And naive machine learning algorithms would basically say, oh, great, this is the lowest mortality risk, so let's push all of the patients into this regime. Instead being able to argue about these discontinuities and then discover a mixture model, which is allowing you to now have these sharp thresholds brought in as indicators of causality of those interventions. So this is now described in a paper that we posted on medRxiv called Death by Round Numbers and Sharp Thresholds that I really encourage you to read up. We've also gone beyond individual tissues and asked how are these global effects, such as is known for exercise, affecting not just one tissue, but many tissues at the same time. So we looked at muscle, we looked at visceral fat and subcutaneous fat, and also small intestine, cortex, liver, and heart and blood. And we looked both in mouse and in human to basically look at what is the impact of a sedentary lifestyle or an exercise lifestyle, and what is the impact of a normal diet versus a high-fat diet. And what we found is that, indeed, the high-fat diet is remodeling the tissue to basically become more lipogenic. So if you look at these adipose tissue, these adipose stem cells, what we see is that they're highly dysregulated based on the impact of this high-fat diet. And if you look at exercise, it remodels the tissue as well, but in exactly the opposite direction. So it is able to restore the beneficial effect of exercise and combat the detrimental effects of a high-fat diet. And we can understand how these are mediated through cell-cell communications and interactions. We can basically predict through ligand-receptor pairs how the signals are traversing between different cell types, and then use those to annotate these functions. We also developed methods for scaling up the perturbation from a single experiment at a time to many more experiments at a time. So we've developed these modular and programmable CRISPR-based constructs that allows to either couple them with Cas9 for genome editing to knock out a promoter or a gene or to edit individual SNPs, as I showed earlier, or with deactivated Cas9, which does not cut the DNA, but instead can be used to recruit inhibitors such as SCRAB or activators such as P300 to then turn on or off individual enhancer regulatory control regions. So we've used these to now manipulate the enhancer of BIN1, for example, and start looking at how we can change its expression to be higher or lower. We've also differentiated cells into neurons, microglia, astrocytes, and oligodendrocytes to understand how these perturbations impact different cell types differently. And in collaboration with my former postdoc, Andreas Penning, who's now a professor at CMU, we're basically looking into introducing individual mutations associated with Alzheimer's disease that we predict to be causal in the brains of mice through these high throughput reporter assays that allows you to test thousands of enhancer variants in a multicellular setting in the brain of an animal. We've also developed these ultra high throughput assays that allows us to carry out 7 million experiments in a single assay. How is that possible? We can basically cut the DNA in accessible regions and then insert these accessible regions in self-transcribing constructs that allows to then measure for every one of these constructs, how much does it drive gene expression in a reporter assay? And that allows us to now find the subset of these constructs that are showing high activity. And then this subset allows us to infer computationally in a very narrow way exactly what are the nucleotides that are responsible. And you can see here that we're predicting these high evolutionary conservation nucleotides that also overlap regulatory motifs. So where do we go from here? What we want to do systematically is develop new therapeutics that allows to now go after these new targets that we are identifying. So in collaboration with Brad Penteluter from the chemistry department and Marinka Zitnik from the medical school, we're basically looking to connect regulators with motifs, with individual variants, with enhancers, with genes, and then specific cell types where they act and the specific proteins. So all of this comes from the work that I showed you so far. And then using the tools that Marinka is building, we can now look into the protein structure and the drug chemical structure in these language models that allows to look at the three-dimensional conformation of these drugs in these pathways, project them into the different spaces of representation for these structures through graph neural networks, and then start connecting them together to understand how we can use those to impact disease. So we're using multiple therapeutic modalities to target IRX3, for example, that I showed you earlier, at the protein level, at the target regulatory site, and at the upstream regulator or the RNA level itself. So I could sit here and tell you how exciting these things are and how we've written a lot of papers that have hundreds of thousands of citations, and we have dozens of grants. But instead, I'm going to leave you with a much darker note that disease is still reigning. We have many predispositions within this room, within my own family, the genetics, the environment, there's systemic disorders, we have pathogens, and the increasing lifespan is basically giving you increased prevalence of Alzheimer's and many new diseases. And we really need to transform pharma. We can't just simply stay with misdiagnosis and only treating the manifestations of disease. And we can't be thinking of disease in a monolithic way with silos that separate different departments. Instead, what we need to do is enable personalized medicine. Use all of these results that I told you about to start understanding the mechanism of disease, to be able to manipulate these mechanisms by reversing the circuits that we uncover, to enable individualized treatments by combining multiple pathways, by developing drugs for every pathway, and then combining these drugs systematically for every patient. For every patient, we have a burden, an accumulation of perturbations for each of the pathways. And then we can use all of the omics, the genetics, the epigenomics, the transcriptomics, the proteomics to understand how these are clustering specific pathways. And we can't do that alone. We need a coalition. We need you guys. We need computer science and engineering students across machine learning and deep neural networks to understand the DNA code, to understand the circuitry, to be able to integrate big data. But we also need collaborations with biology, with chemistry, with biotechnology. We also need partners in finance to enable long-term vision in these developments from pharma, from patients and hospitals. So to enable all this, we basically started this new coalition with Brad and Marinka that I mentioned earlier, that allows us to now look at these systematic predictions that we're making about the driver genes and domains, to be able to use these high throughput experiments that I showed you earlier, and to use these geometric deep learning approaches to guide both disease and gene prioritization, and leverage these extremely high throughput, extremely massively parallel and multimodal ways of synthesizing these diverse therapeutic modalities. And what we're doing is now, in collaboration with Marinka Zitnik and several students that are co-advised by the two of us, we're basically bridging this geometric deep learning together with large language models and self-supervised learning of individual molecular interactions to effectively project into the same space protein structure, drugs and molecules, biological knowledge from BioGPT and MedGPT, general knowledge from GPT-4, medical events from electronic health records that I showed you earlier in Mimic, functional genomics from the single-cell RNA and the single-cell attack data that I showed you earlier, the variation in a population, both genetic and phenotypic through the UK Biobank, case control genetics through the genome-wide association studies, and the systematic perturbation experiments through Perturb-seq and L1000, through contrastive learnings to learn these multimodal joint knowledge projection embeddings that allow us to now connect all of these different types of language and understanding to be able to speak across the different modalities. More generally, we're seeking to navigate this seascape of biological knowledge. We're basically using AI for science, building new collaboration tools for human AI partnerships with explicit representations and navigations of this knowledge space. So for those of you who are heavily computational, who are interested in deep learning and machine learning, please reach out, because we're looking for help both in this project and in that project to be able to sort of project all of these complex information into the same space. So again, we're looking for postdocs, we're looking for students. For those of you graduating who want to carry out your studies at MIT, please join us. So this is our website, compbio.mit.edu. Again, my name is Manolis Kéllis. You can mail me at manoli@mit.edu. And I would be thrilled to work with you to integrate big data and machine learning and systematic experimental validation to both have a lot of impact and a lot of fun. And again, I'm not alone in this. There's a lot of professors at MIT within computer science that are working in the medical space, in the neuroscience space, in genomics, in proteomics, in synthetic biology, vision. And we are only one institution here, the Computer Science and Artificial Intelligence Laboratory, but we are surrounded by this ecosystem of biotech, of AI, of machine learning, of computer science and algorithms. So that's where I'll stop. And I will be truly delighted to talk to many of you. And again, I'm so sorry I'm not going to be able to be there. It's going to be 3 a.m. my time when the talk is scheduled. But I hope that you will be able to watch this recording in a couple of hours. Ευχαριστώ και πάλι πάρα πολύ και πραγματικά μεγάλη μου τιμή που μιλάω σε εσάς και σας ευχαριστώ πάρα πολύ για την όμορφη πρόσκληση. Γεια σας.